welcome to Bosons Botany. In this video, I will be defining glycolysis. And glycolysis is a very important pathway or metabolic pathway, and it is the first metabolic step. Glycolysis, then uh, your Krebs cycle, then ETS, that is electron transport chain. These three important metabolic pathways are very important and carried in a very sequential manner. So glycolysis is the first metabolic pathway. As you can see, this image has been taken from the book Leninger, and all the 10 steps are very clearly mentioned over here, along with the enzymes and the intermediates, along with the final product, which is pyruvate, showing ATP production and ATP generation. But I think this is little bit confusing for beginners because it is little difficult to remember all the steps along with their enzymes. So for your convenience, I have created a table, or you can say tabular explanation, to understand and remember glycolysis very easily. So let's begin. I have divided glycolysis into three main phases: energy investment phase or priming stage, splitting phase, and energy generation phase. One by one, I will be dealing all the three phases along with the products, the enzymes, and few important points, and also ATP generation and utilization. Now, the first step, which is the energy investment phase or priming stage, is clearly defining the usage of energy, which is ATP. Now, the first step in this phase or the first reaction in glycolysis is glucose 6-phosphate converting into glucose 6-phosphate. As you can see, there is addition of phosphate on the 6th carbon of glucose. Therefore, therefore the reaction is known as phosphorylation, and glucose is a 6-carbon compound. The second step of glycolysis is the conversion of glucose 6-phosphate into fructose 6-phosphate. And I think it is very clearly visible. I have used two arrows here to show bidirectionality or the reversibility of this reaction. That is, this can be reversed, but the first step cannot be reversed. It is pointing towards the product side. But in the second step, it is bidirectional. And this step is isomerization because glucose 6-phosphate and fructose 6-phosphate are isomers of each other. And fructose 6-phosphate is also a 6-carbon compound. The third step of glycolysis is the conversion of fructose 6-phosphate into fructose 1,6-bisphosphate. And this is again an irreversible reaction, showing only one arrow towards the product side. And fructose 1,6-bisphosphate is also a 6-carbon compound. Now, the energy consumption or energy production, as already I have told you, in energy investment phase, it is definitely using energy but not producing energy. So always remember, in glycolysis, the first three steps, that is glucose converting to glucose 6-phosphate, glucose 6-phosphate converting to fructose 6-phosphate, and fructose 6-phosphate converting into fructose 1,6-bisphosphate, will never ever generate ATP or energy, but they will use energy or ATP. Okay, so energy is used up here, and how many energy or how many ATP molecules? 2 ATP molecules are used up. And where they are used? In the first step, that is glucose to glucose 6-phosphate, 1 ATP is used up. ATP converting into ADP, right? In the second step, that is the reversible phase of glycolysis, no ATP is used up, that is nil ATP molecule. In the third step, again 1 ATP is used up. So in total 2 molecules of ATP are used up. And it is very clear from these two steps that I have highlighted here. The first step where one phosphate is added, and in the third step where another phosphate is added, means from ATP phosphate is added. Okay, and therefore energy is used up in first and the third step. But in the second step it is only the isomerization, that is conversion of one isomer into other. So no addition or removal of phosphate, it is only the conversion product. But in the first and the third step phosphates are added, that is why energy is used up in first and the third step. So total two molecules are used up, where in glucose to glucose 6-phosphate and fructose 6-phosphate to fructose 1,6-bisphosphate. I hope this is clear. I'm going little slow so that I can explain you glycolysis with some important key points. Okay, so have patience and try to understand. Now, what are the enzymes and cofactors needed here? In the first step, that is glucose to glucose 6-phosphate, hexokinase is used, and cofactor is magnesium plus 2, that is magnesium ion. In the second step, phosphohexose isomerase is used. Why isomerase? Because it is the isomerization process. And here also magnesium ion is used as a cofactor. In the third step, which is fructose 6-phosphate to fructose 1,6-bisphosphate, phosphofructokinase 1 is used. Let me tell you, hexokinase and phosphofructokinase 1, these two are very important enzymes. Why? I will tell you later on. Now the inhibitors. Inhibitors are like for example glucose 6-phosphate, glucagon. These are inhibitors of hexokinase, and these will inhibit the first step, that is glucose to glucose 6-phosphate. And let me clarify one thing here. Why glycolysis is carried out in our body? This is a very basic question that might come to your mind, right? Why we study glycolysis, why it is happening, and why it is needed. So, glycolysis, it is basically conversion of glucose into pyruvic acid. That is, glucose is broken down in a simpler uh, organic compound, which is the pyruvic acid. Now, if glucose is very less in your body, okay, glucose is very less in your body, and you need to have ample of glucose so that you can do your activities. Now if glucose is less, what will happen? Glycolysis will be inhibited. Why? Because glycolysis happens when glucose is in excess, when glucose concentration is high in your blood. When blood glucose level is high, at that moment glycolysis will operate, because excess amount of glucose should be broken down into smaller organic compounds like pyruvic acid, acetyl coenzyme A, and many more intermediate products. But when glucose level is low in your blood, then why we need glycolysis? It should be inhibited. And if we inhibit the first step of glycolysis, then the next 9 steps will not occur. They will be stopped. They will not at all occur. So, the first inhibitors of glycolysis are glucose 6-phosphate and glucagon. And if you have any idea about glucagon, you can definitely get like the use of glucagon. You can understand the use of glucagon. But if you don't know about glucagon, let me tell you, glucagon is an enzyme that is released from the beta cells of pancreas, and glucagon basically uh, functions when blood glucose level is less in your body. But insulin, if you know insulin, it is released from the alpha cell of pancreas. Insulin is released when blood glucose level is high. So glucagon is released when blood glucose level is low, and this will act as an inhibitor for the first step of glycolysis to inhibit glycolysis process. The second step does not have any inhibitor, and the third step is having ATP, citrate, and hydronium ion, or precisely low pH condition, as the potent inhibitors. Okay, now let me go little bit into deep concept why ATP, citrate, hydronium ion, or low pH is acting as an inhibitor for the third step. Because in the third step, ATP is used. Now, if ATP itself is inhibiting the process or the step, then the step will not happen. And citrate, low pH, why they are acting as inhibitors? Because citrate and low pH, they are uh, or precisely citrate or citric acid. It is the intermediate product in case of Krebs cycle, if you know, or TCA cycle, that is tricarboxylic acid cycle, which happens just after glycolysis, which is the second important metabolic step in our body. So in Krebs cycle, citric acid or citrate is produced, and if citrate is high in your body, means citric acid means low pH, acidic condition means low pH, means high hydronium ion. These things are actually telling the body, the cells, that we are high in energy. We don't require any more energy. We don't need the process of glycolysis to happen, because in glycolysis we get pyruvic acid along with ATP also as energy. But if energy is excess, if there is high amount of hydronium ion, high concentration of citrate, high concentration of ATP, then why we need extra amount of energy? So these will act as inhibitors for the third step. I hope it is clear. Now the activators. Activators are definitely the opposite of inhibitors. So if you remember inhibitors, you can automatically remember the activators also. So the activators for the first step are AMP, that is adenosine monophosphate, insulin. Why? Because now glucose, blood glucose level is high, and when blood glucose level is high, definitely glycolysis should occur, because we need to break down glucose, excess glucose, and for that we need insulin. And also ATP is less, or energy is less, so we need AMP, that is adenosine monophosphate, to tell the first step that okay, you are ready to be further processed and you can create pyruvic acid as the end product. So we are allowing you to carry out glycolysis. Second step again does not have any activator, and the third step has fructose 2,6 bisphosphate, AMP, inorganic phosphate, and high pH, or you can also say ammonium ion, as the activators. So these are very conceptual things. If you remember the concept behind the need for glycolysis, you can understand the need for these inhibitors and activators. Now some important points regarding enzymes which are involved in these steps and the intermediates which are produced. So first is the enzyme hexokinase. In all tissue it is present, and it is having low Km value. What is Km? Km is the dissociation constant. If dissociation constant is low, means The enzyme has high affinity for glucose, means it will be highly bound to glucose molecule because the dissociation constant is less. It will not dissociate from glucose. It will very tightly bind with glucose. And hexokinase has low Km value, that is low dissociation constant value. That is why it has very high affinity towards D-glucose and hence uses glucose at low concentration also, because if you can understand this line, low Km high affinity for glucose means if you add few concentration or few amount of glucose, then also hexokinase will easily bind with glucose. It does not require high concentration of glucose because it has high affinity towards it, right? It is a soluble enzyme. It catalyzes the phosphorylation of various hexoses, and it is an allosteric enzyme. And there are total 4 types of hexokinase enzymes. Now, what is an allosteric enzyme? Very briefly, allosteric enzymes have multi-subunits, or you can say they are multi-subunit complexes, and they give you a sigmoidal curve. Okay, they give you a sigmoidal curve, and they are multi-subunit complex enzymes or proteins, because enzymes are proteins. So hexokinase, it is an allosteric enzyme. Now glucokinase. Glucokinase, it is found only in liver, and it is present in the nucleus when not working. Means when it is inactive, it is present in the nucleus of the liver cell. It is just opposite of hexokinase, means it will have high Km value, that is high dissociation constant, means it has low affinity towards D-glucose. It catalyzes not all hexoses. Unlike hexokinase, it will not catalyze all the hexose molecules. And it is a Michaelis Menten enzyme, means it has a hyperbolic curve. It is not having a sigmoidal curve, and it is having single subunit. Isoenzyme of hexokinase, and as I have already mentioned here, there are total 4 hexokinase. Okay, so again I have written here the same thing. So it is an isoenzyme of hexokinase. Now glucose 6-phosphate, that is the first product of glycolysis, glucose 6-phosphate. Glucose 6-phosphate is impermeable to the cell membrane. It is the central molecule with a variety of metabolic fates: glycolysis, gluconeogenesis, glycogenesis, and pentose phosphate pathway. Means glucose 6-phosphate is used as an intermediate in all these metabolic steps. Now, phosphofructokinase. It is an allosteric enzyme. Again, it will have a sigmoidal curve. It is multi-subunit complex, the activity of which is controlled by several allosteric modulators. This catalyzes the rate determining step. Means this is the rate determining enzyme, and hence it is very important. So I have completed the first column here, which is energy investment or priming stage, where 2 molecules of ATP are used. Now the second column, which is the splitting phase. Splitting phase means something will split into 2 or more than 2 compounds. Let's see. The reaction here is fructose 1,6-bisphosphate. See, fructose 1,6-bisphosphate is the final product in the priming stage. So this product will come here in the splitting phase. So fructose 1,6-bisphosphate, it is a 3-carbon compound. Sorry, it is not a 3-carbon compound, it is a 6-carbon compound, but it will be converted into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. And these 2 molecules are 3-carbon compound, right? So fructose 1,6-bisphosphate is converted or broken down or splitted into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, and these two are 3-carbon compound. Now dihydroxyacetone phosphate, it will be converted into two molecules of glyceraldehyde 3-phosphate. Remember this step. Okay, remember this step. In the first step, what we are getting from fructose 1,6-bisphosphate, we are getting glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, and dihydroxyacetone phosphate will convert itself into two molecules of glyceraldehyde 3-phosphate. Always remember to keep the valency same. Valency means if fructose 1,6-bisphosphate was 6 carbon compound, then the final products or the final molecules should also have 6 carbon compounds. Okay, so these two are 3 carbon compound, that is glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So 3 plus 3, 6 carbon compound, and dihydroxyacetone phosphate, which is a 3 carbon compound, means it will produce two molecules of glyceraldehyde 3-phosphate, in total making 6 carbon compound. And here we are using no energy, so no problem at all. And what are the enzymes and cofactors used? In the first step, we are using aldolase having zinc as a cofactor, and in the second step we use triose phosphate isomerase. Okay, it is also an isomerization process. And inhibitors, no inhibitors for the first step, and for the second step, that is conversion of dihydroxyacetone phosphate into two molecules of glyceraldehyde 3-phosphate, the inhibitors are bromohydroxyl acetone phosphate and glycidol phosphate. Activators, no activators. And points, I have already explained you here, but again let me read it for you so that it gets clearer. Why two molecules of glyceraldehyde 3-phosphate are made from one glucose molecule? Answer is fructose 1,6-bisphosphate makes one molecule each of glyceraldehyde 3-phosphate and DHAP, that is dihydroxyacetone phosphate, both 3 carbon. Glyceraldehyde 3-phosphate will enter into the next step, but DHAP will not, but it will convert into glyceraldehyde 3-phosphate. So after splitting phase, two molecules of glyceraldehyde 3-phosphate will be carried forward, and so everything will become twice after this phase. So this phase is very important because DHAP is ultimately converting into two molecules of glyceraldehyde 3-phosphate, and this will actually work for further steps, not DHAP. And from fructose 1,6-bisphosphate, carbon 1, 2, 3 will form DHAP, and carbon 4, 5, 6 from fructose 1,6-bisphosphate will form glyceraldehyde 3-phosphate. Now let's get into our last step, that is the energy generation phase. Remember one thing, the first column here, that is the priming stage, and the third column, that is the energy generation phase, these two columns are basically important columns. If you forget about splitting phase but remember only the main concepts that I have already highlighted here, then it will help you. Otherwise you need to remember the priming phase and the energy generation phase. So let's start. Energy generation phase means now energy will be produced, means ATP will be generated, we will get our ATP currency. Now the first step here is glyceraldehyde 3-phosphate, which is already derived during the splitting phase. As I have already told you, DHAP will convert into two molecules of glyceraldehyde 3-phosphate. Now this glyceraldehyde 3-phosphate, that is one molecule, okay, I'm considering one molecule first. One molecule of glyceraldehyde 3-phosphate will convert into 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate. And this is again a reversible reaction. 1,3-bisphosphoglycerate will convert into 3-phosphoglycerate. This is again a interconvertible reaction. 3-phosphoglycerate will convert into 2-phosphoglycerate along with one molecule of H2O, that is water. Again, reversible reaction. 2-phosphoglycerate converting into phosphoenol pyruvate, that is PEP, which is again reversible, and PEP will convert into pyruvate, and this is irreversible in nature. Now just consider another molecule of glyceraldehyde 3-phosphate and repeat these steps. So we will get 2 molecules of pyruvate from 2 molecules of glyceraldehyde 3-phosphate. I hope this is clear. I have written here all 2 molecules. Okay, just take one molecule and repeat it for the second molecule, and you will get 2 molecules of pyruvate, which is our end product or the final product of glycolysis. Now energy, 2 NADH plus 2H plus used in the first step, that is glyceraldehyde 3-phosphate converting into 1,3-bisphosphoglycerate. 2 molecules are generated. 2 molecules are generated in the second step, that is 1,3-bisphosphoglycerate converting into 3-phosphoglycerate, 2 molecules of ATP. And in the last step, that is PEP converting into pyruvate, 2 molecules are generated, not used, generated. Again, let me tell you, in terms of energy, we are getting 2 molecules of NADH in the first step, 2 molecules of ATP in the second step, and 2 molecules of ATP in the last step, that is PEP converting into pyruvate. So in total we are getting 4 molecules of ATP, right? 4 molecules of ATP and 2 molecules of NADPH. Now the enzymes and cofactors. The first enzyme which is written here is glyceraldehyde 3-phosphate dehydrogenase, which is used for the first step, and it is having cysteine at the active site, and it is the oxidative stage. Okay, it is the oxidative stage. And always remember one thing, when you see dehydrogenase, then it is having NADH or FADH as the energy currency. So here it is dehydrogenase, means we have NADH, and this is for the first step here. And this is the oxidative stage, and this enzyme has cysteine at the active site. The second step, which is 1,3-bisphosphoglycerate to 3-phosphoglycerate, the enzyme is phosphoglycerate kinase, and this kinase will be showing reversible reaction. Now what does this mean? Actually all the kinase mediated reactions are irreversible in nature. All the kinase mediated reactions or steps are not interconvertible, they are irreversible. But phosphoglycerate kinase is an exception here. Being a kinase, it will show a reversible reaction, that is interconvertible reaction, and I have highlighted here as you can see so that you remember this step and the enzyme used here. Now the third step is having mutase as the enzyme, and it has histidine at the active site. The fourth step is enolase, which is catalyzed by the enzyme enolase, and the final step, it is catalyzed by pyruvate kinase, and it is having cofactors like manganese, magnesium and potassium, and inhibitors like iodoacetate and arsenate. Iodoacetate and arsenate will inhibit the enzyme phosphoglycerate kinase. Iodoacetate and arsenate will inhibit phosphoglycerate kinase, and ATP alanine will inhibit pyruvate kinase. Activators AMP, fructose 1,6-bisphosphate for pyruvate kinase. And some important points. First step is the oxidation and phosphorylation. Second step And the last steps are substrate level phosphorylation. Second and the last means this second and the last. Why? Because we are getting ATP molecule, and the reactions which are giving you ATP means they are substrate level phosphorylation steps. Now, in the priming phase, we have used up 2 molecules of ATP, and in the energy generation phase, we have generated 4 molecules of ATP. So, the net gain of ATP in glycolysis is 2 molecules of ATP and 2 molecules of NADPH produced, as you can see here, 2 molecules of NADH. I hope it is clear, and here it is the final reaction. Glucose plus 2 NAD plus 2 ADP plus 2 inorganic phosphate will make 2 pyruvate, 2 NADH, 2 H plus, 2 ATP and 2 water molecules. It is very important you remember this final reaction, and the regulatory steps are catalyzed by hexokinase or glucokinase, fructose 1, phosphofructokinase 1 and pyruvate kinase, means they will regulate the main regulatory steps. Now the last few points. Key points: kinase means addition of phosphate group, may or may not be substrate level phosphorylation. Reaction catalyzed by kinase are irreversible, but exception here phosphoglycerate kinase. Okay, it is the exception. And dehydrogenation reaction means it will have NAD plus or FAD plus to generate NADH or FADH. Now there are few more points. Glycolysis, it is universal step referred to as Embeden-Meyerhof pathway, EMP pathway. Glycolysis is defined as the sequence of reactions converting glucose or glycogen to pyruvate or lactate. Lactate is generated in absence of oxygen, that is anaerobic glycolysis, and pyruvate, which I have defined, it is aerobic form of glycolysis, that is in presence of oxygen, with the production of ATP, and enzymes are produced in the cytosol, means glycolysis happens in the cytosol. Glycolysis occurs in absence of oxygen, as I have already told you, anaerobic, or in presence of oxygen, aerobic. Lactate is the end product under anaerobic condition. In aerobic condition, pyruvate is formed, which is then oxidized to CO2 and water. Glycolysis is the major pathway for ATP synthesis in tissue lacking mitochondria. Example: erythrocyte, cornea, lens, and sperms. So the cells which are lacking mitochondria will generate ATP using glycolysis, because mitochondria is the powerhouse of the cell, as we all know, generating maximum amount of energy. But if in case some cells do not have mitochondria, like our red blood cells, cornea, that is cells in the eyes, and lens cells, then sperms, they do not have mitochondria, so ATP is generated using glycolysis. Glycolysis is very essential for brain, which is dependent on glucose for energy. The glucose in brain has to undergo glycolysis before it is oxidized to CO2, water. Glycolysis and water. Glucose plus 2 ADP, 2 inorganic phosphate, will give 2 lactate and 2 ATP, which is the anaerobic glycolysis. I have written here anaerobic glycolysis. And intermediates formed in glycolysis are useful for the synthesis of non-essential amino acids and glycerol. And glycerol is used for fat formation. So I hope this is clear, and I have tried to give you an overview of glycolysis without actually defining this conventional figure which we all study in our school, in our colleges, because this is what we are like taught, or we are said to remember. But there are ways to remember things, and I have tried to make you understand in a very easy way. but I would suggest you to please go through this figure at least once, because it is again important for your school exam. If you are told to write the steps in a definite flowchart manner, then you should understand this image as well. But it is not difficult once you have understood my tabular explanation. So I hope this will help you. Thank you.